Hi, welcome to the ZP Developer Zone. So every Thursday at 8 a.m. London time, we like to do this webinar for the members of our ZP Developer Zone where we answer questions that have come in, regarding well, basically technical questions. Now it says on my slides here that it's the 28th of July, 28th of July, and I've saying it's 8 a.m. But in fact, I've actually started this webinar early. Now, the reason being is, and I'll sort of cover that in the first part here, is this week we're actually at the AAC in we're at the AAC in Chicago, and I'm having to do this early because of all the different time zones differences. I need to essentially do this webinar a little bit early. So sort of my apologies for that, but I will essentially carry on. And like I say, we do like to do this live, but this week I'm in Chicago. It's in the US. It's a different time zone. And we've got a lot of commitments on the stand, so I've essentially started early. So let's carry on anyway. So we have our the slides. If you're new to biosensing and electrochemistry, then obviously try out the ZP Academy. There's a couple of free courses, and I know that some of the people who are attending the webinar or will be following the webinar this week have already done that. We obviously have our webinars, we have our collaborations, we also have job postings, and we have our ZP Developer Zone website. And we also have workshops, and it's probably just worth saying, I mean, I think this workshop is people who really want to get into biosensors. We have a workshop running in September in Norway, and I'll put a link here. Now, any sort of links that sort of pop up during the webinar, you can essentially feel free to, I'll put those links underneath the video when I finished. And I will go a little bit faster because I do have about 40 odd things or slides today. So first of all is we had an inquiry regarding the principles of glucose sensing. I will talk quite fast during that because I do think we've covered this quite a bit. But I'll maybe talk about open source potentiostats, which might be more relevant to this inquirer. We have a question around the measuring osmolarity for dry eye. We have I think this must be my favorite question of the week, measuring the pH in cattle for 5 years, and then also sensors in the biopharmaceutical industry, and then also we use this forum to sometimes answer questions that come in from people regarding things like our Hypervalue electrodes. So we have a couple of sort of questions around that. So let me go a bit faster. So question number one was this person's, and I appreciate this, has actually already watched our free course on the ZP Academy on biosensors, and he would like to use our sensors in his project. It's probably worth saying at Zimmer Peacock, obviously, we have a whole bunch of sensors, including glucose sensors. I think this particular person is interested in doing a non-invasive wearable sensor. I'll discuss the non-invasive slightly in a bit, but our sensors can lend themselves to being wearable. We do have a whole platform where we can take our sort of wearable sensors, turn them into a sort of a rapid prototype, and using the connectivity to an app, which we obviously have app development, and then we have this Julie. Julie will appear a lot. It's a proprietary database. It sits in the cloud. It can receive electrochemical data, and that electrochemical data could be glucose or pH or lactate, and then we can essentially do calibration up in the cloud and give data. Sending data to Julie, especially things like glucose data, is something that we've already done. So it's more than feasible. It is possible, and it has been done. Now, the background to this is the person really wants to know, I think they want to know things like the kind of voltages you apply. So I know I've done these slides in the past, and I sort of apologize for those regulars. People like Aftab, who will be watching later on, will recognize all this, that you can have a sort of an electrode, you have a mediator, you have an enzyme layer, and you have a barrier layer. So that's a typical sort of, that's one way of making what we call a generation 2 glucose sensor. Just 3 stacks of materials. You have the electrode on the bottom, a mediator on top of the electrode, the enzyme layer, and then the barrier layer. The job of the barrier layer is to keep not all is to prevent not all is to prevent some of the glucose actually getting to the enzyme. In the enzyme, we have a sort of an enzymatic reaction, but the important part really is that you get a molecule called hydrogen peroxide. Once you've got hydrogen peroxide, electrochemists like hydrogen peroxide, it gives us a sort of handle on which we can then detect. So hydrogen peroxide can be detected using a mediator. Now this is the important bit. If you're using a mediated electrode, you might be using a voltage of sort of 0 to 200 millivolts. So if you're designing electronics for a generation 2 sensor, then the voltage difference between your reference and working might be 200 millivolts. I am going to link to some open source potentiostats in a bit. And that, if you're coming from an electronics perspective, whereas I'm sitting here talking about chemistry, I think coming from looking at open source potentiostats might be a good way of you understanding what you need to do. I always advise people really, if you want to get it into glucose sensing, I would, not always, but I would suggest what we call a generation 1 glucose sensor, where we get away with that. We go away from the mediator and we just detect the hydrogen peroxide directly. And there we use a voltage of 650 millivolts. So I suppose I've given you the idea that if you're getting into sensing or glucose sensing, there's at least 2 versions of glucose sensor or electrochemical glucose sensors, generation 1 and generation 2. Generation 1 use 650 millivolts. Generation 2, the reason I sort of say 0 to 200 millivolts depends on the mediator that you're actually using. So keep it simple, stay on the generation 1 glucose sensor. Here's some background information. I've done webinars on this in the past, but there's something called the seed stat out there. The only reason I'm recommending the seed stat as a basis for designing electronics is because the guy who does this, he seems to be currently supporting it. With open source materials, sometimes the inventor or the original developer kind of comes on and does a good job, but then they sort of drift away from the project, and essentially the project ages, things become unavailable, and but with the seed stat, I know this is quite a current project. I've spoken to the guy online, with a few messages, so I know he's active. So I'd say the seed stat is probably a good open source potentiostat to take a look at. As per normal, I'll put a link underneath the video so you can find it. The guy said to us in his inquiry, I'm interested in non-invasive glucose sensing. So it's just for your interest, there's a free ebook out there called Pursuit of Non-Invasive Glucose, Hunting the Deceitful Turkey. So I just, as a sort of recommendation, I would actually have a look at that. So like I say, if you're watching this and thinking, I often do this live. I'm actually live streaming at the moment. I've just moved the live streaming forward a bit because, as I say, we're at the AACC and just really busy, different time zone. So to make things easier, I've actually done this live. Now, the problem I have is with this particular inquiry is the gentleman would like some glucose sensors off us, and we're very happy to get them to him. The problem is that he's based in Turkey or Turkey, and the problem is that there's so many import tariffs into Turkey. Even if you try to, for example, send people samples or send them for free, they're going to end up with a lot of import tariffs. So I say in this regard, if you're in Turkey and you want ZP products, even if you want some free samples, you're going to have to contact a company called Biosensor. They're our distributor in Turkey. And it's just simply because I understand this gentleman's, a high school student. I respect that. That's why we're doing it in the presentation. But it should know that even free is hard because of the import tariffs and the complexity of importing into Turkey. So my apologies for that. But I hope, we do like your inquiry, and we have, tried to sincerely kind of cover it in this webinar. Now, question number 2 was really discussing measuring osmolarity for dry eye and also intraocular pressure. I'm actually going to focus on osmolarity. I realize that actually I could have also discussed intraocular pressure as well, but let me focus on osmolarity today for measuring dry eye. So I think that obviously the inquirer already knows this, but for anyone else's interest, we do have a note about measuring osmolarity and how osmolarity, it has been clinically proven to be proportional or a clinical indicator of dry eye. Osmolarity is a sort of number of electrolytes per liter of sample. Now, I'm not expecting, obviously, people are not going to give up a liter of eye fluid, but the units osmolarity may be not so familiar to people, but it's really the amount of electrolytes per volume. And really, the higher it is, then essentially the more sort of acute that dry eye is. So you can see that we kind of have a sort of normal at around 300 milli os up to about 340. So dry eye, it's an indicator of amount of electrolyte in the volume, and you can measure this kind of material through conductivity. It's a side note, but osmolarity also appears sometimes in measuring the hydration in athletes. So osmolarity is a kind of interesting one, and we do have some notes on the website about that as well. So if you're going to measure osmolarity as an indicator for dry eye, you probably need a sensor, and if you get the design right, the fundamental sensor for this could be much, much less than $1 per sensor. But you do have to pay the NRE, you do have to pay for the research and development. You know, getting $1 essentially doesn't come for free. You do have to put in the sort of the effort in order to get to something like a $1. But the fundamentals of manufacturing these kind of sensors is actually low cost. But on top of that sensor, you're also going to need some microfluidics as well. Now, the microfluidics will stack some costs onto this. It's still possible to get this for less than $1. But again, you do have to put in the R&D on this. You do have to pay for the sort of manufacturing and the validation and stuff. But if you want to keep simple and just measure osmolarity, then you can do it in a microfluidic device at actually quite low cost. Now I will, and then the last thing you're going to need is electromechanical. How am I actually going to hold and use this device? Now we do have something called the OmniSense. The OmniSense itself, its housing is not suitable for this application, but, and I will touch upon it, it can be made a lot smaller in a minute. So if you see a link, then I'll put the links underneath the video afterwards. Like I say, this is unusual that we're actually streaming earlier than we said we would, and like I say, it's because we're at the AACC this week. Now, if you're trying to measure dry eye using osmolarity, you could get to markets. Now, this is an opinion, and I'd have to really talk to my my RAQA team, our regulatory assurance, quality assurance team. But the development and the sort of clinical validation would be a bit like this: that you'd have to develop a osmolarity sensor with a microfluidic chamber upon it, so that you could sort of essentially capillary fill some tear film from the eye, and you'd have to have a meter. Now the thing is, the reason I bring in this shot of the finger is the electronics that you actually need for this are actually intrinsically small. They're the size of the end of my finger. So don't let the big bulkiness of this OmniSense make you think, oh no, it can't be shrunk. No, it can be really shrunk down. Now what makes it a little bit easier to do the at least initial or preclinical validation on this is there's actually a predicative device. Somebody else has already done this, and therefore you may, and this has to be discussed as part of a consultancy with a, for example, our team is adept at this kind of thing. You have to talk about your clinical path. Can I do a 510K? Because a 510K in the USA would allow you to compare yourself against the device that's already on the market. And if you can prove an equivalence, then the FDA will give you a lower cost route to market than pretending it's not pretending, but it's saying it's a new device and you're having to do a lot more testing and validation. So there's a predicative device out there. It will give you the truth. It will, that this has been FDA approved. Now for us, we can potentially develop the sensor, put the microfluidics on it, use our OmniSense, send our data via our app to our database called Julie, and in Julie then we have the raw signal. So what Julie allows us to do then is gather raw signal potentially from volunteers and actually look at the signal, but then compare the signal versus the device that's already on the market. So this is both a way of validating what we're doing is giving, let's say, clinically correct answers, but also by comparing it to the device that's already on the market, you're also preparing the documentation for a 510(k) submission. And what we do then is we um, we calibrate ourselves against the device that's already in the market, and then we have to essentially verify and validate that it actually is accurate and precise enough. But if you're trying to make a sensor for measuring osmolarity and using that as a predictor for dry eye, your life is made easier because somebody's already done this. So it makes your life easier because you actually have a device to both compare against in development, and you also have a device to compare against when trying to do regulatory approval. And I know in the US, I think they call not think I know they call it a 510(k), and it can actually offer you a lot lower cost to market and a lot less risk to market. Now the cost to get to market, ZP has covered this. Again, if you see a link, I have covered the cost of getting, essentially this is a form of IVD, an in vitro diagnostic, and we have covered the cost of this in videos and on web pages. Now the predicative device in this case, it costs them, now this is off the top of my head because I was writing this presentation on a plane and I wasn't able to watch the video, but I think it cost them about $7 million US, and that was to get regulatory approval. Now, I think, this is my opinion based on sort of reading around, that 2/3 of their cost was actually getting the sort of regulatory assurance, quality assurance part actually done. And I then put a sort of bullet point here about the ZP effect. Is the ZP effect is this, that we've done these kind of things a lot in the past, and so it just when ZP is doing this kind of stuff, we often end up doing it at a lot less cost than otherwise it would have necessarily taken people. So that's $7 million US. As I say, 2/3 of it was actually on the regulatory side. Now the regulatory side is actually easier now because somebody has already gone through the regulatory side of it. This company TLab. So these are the historical numbers. It's good to have those in mind when you're thinking about developing products. But if you're following after somebody else and you're going to go the 510(k) route, you might be able to get there. And it depends really on the efficiency and the quality of your team and the efficiency and the quality of the consultants that you hire, etc. But you potentially do it for less than that. But a lot of that depends on the companies themselves. Now, I've said this already, but at Zimmer Peacock, for example, we do have an RAQA team, and they would have to look at whether a 510(k) was the right strategy. I certainly wouldn't make that claim here. I've indicated I think it is, but I think it needs to be looked at by a wider team. For Zimmer Peacock to get involved in programs like this, we call them proof of principles, and that's just the first starting point for us. It's probably worth saying that we do a lot of this sort of development work, and we have some examples of our kind of materials on the website if anyone's interested. I do want to skip forward a bit because I want to talk about science and technology for people, so let me do that. I think this is my favorite inquiry of the week. So somebody wants to measure pH inside the rumen of cattle. Rumen is part of the sort of stomach, and they want it to be there for 4 to 5 years without calibration. So I will comment upon all these points now. I had to Google this myself just to make sure that I knew where the rumen was. So you sort of have the alimentary canal from the mouth to the anus, and then the rumen is sort of one part of the digestive system. I think it's a large sort of part of the stomach, or one of the stomachs. We do have a technology with the right kind of form factor at ZP. I know this video is a bit, this image is a bit small, but there's a length scale along here which is 10 millimeters. So I got a suspicion that we do have a platform that will allow somebody to at least get a pH sensor into the gut of a cow, and we could even send the data out, because I don't want to overstate on this because there's a few things that are concern me here. In this device we have a couple of batteries, there really has to be a risk assessment on those batteries, you know, because you don't want to cause harm to the cow, so that has to be taken into consideration. Um, I will talk about my concerns in a minute, actually, but I think we do have a package that, at least in principle, could work, but there's definitely a lot of details that need to be essentially sorted out on this. Let me just check something just very quickly. Like I said, it's a bit unusual today because I'm usually streaming from, you know, from the office. Today it's a hotel room, and so it's all a bit harder to do this. But let's carry on anyway. Just out of interest, we are very interested in this continuous monitoring applications, both in agriculture and in veterinary applications as well. And we have, you know, just give you a sense of scale, you know, we have been exploring some unusual applications for some of our platforms. The 5-year battery life. I think the 5-year battery life is achievable, but we've never had to run on one of our devices for 5 years. So some engineering has to go around that, around really what you call the duty cycle. You know, how often do you turn on and off? How often do you transmit? Often the biggest drain on battery is not the sensors, it's actually the data transmission. You know, so obviously if you transmit every minute, that's one thing, every second, certainly another thing. But if you transmit every hour, that's another thing. If you transmit, you know, once in the morning, once in the afternoon, or, you know, etc., then that's a very different thing. So I think it's really the transmission of data that's the biggest drain on the battery. My biggest concern, this is actually my biggest concern, is I'm not a veterinarian. How do you keep the sensor in the rumen? Because the whole, you know, physiology of a cow is, you know, it eats at the back end and, you know, passes feces out the, sorry, eats at the front end and passes feces out the back end. Things constantly travel through the cow. So I find it struggle to think how this is going to be actually kept in place. But I'll leave that to the inquirer, because I feel like, you know, you give it to the cow, we can get it sort of, you know, into the mouth and down the I want to say the esophagus, that's correct, and into the stomachs. But then it's just going to carry on its way and actually come out in the feces later on. So that'll be a challenge. And never calibrated. I mean, running a sensor for 5 years. Um, I haven't I haven't looked into if anyone's ever run a pH sensor for 5 years, but I think that's definitely a challenge. Um, there's a low cost way of at least trying this out, and I think the low cost way of this is, I'm biased, but you know, you can buy pH sensors from Zimmer and Peacock. They have the right form factor for what you're trying to do. I think I've put a part number up here, which I think is most relevant. The reason being is the stomach is quite acidic, so I've put in a part number that I think will be resistive, but honestly, you have to try it. I mean, I can't say, I can say we've never stuck a pH sensor in the room of a cow. Your lowest cost option on electronics are, if you Google measuring pH with Arduino, you will find a lot of insights in how to measure pH sensors. We do also have an OEM solution that you can also find. And again, any links, I will remember to put them underneath the video afterwards as well. So your lowest cost option is actually just buy some pH sensors and start using things like Arduinos. The higher cost option is actually to work with companies like us. I just feel there's a risk here, and I think the risk is actually doing this for things like 5 years. That's a risk. Never calibrating for 5 years. I think that's the biggest risk I view on this. Again, any links, and I will put them underneath the video. Question number 4. So I think question number 4 literally came to me whilst I was on a flight. So I tried to do my best with the internet that I had at the time to put together a few slides around this. So they're interested in measuring pH in upstream and downstream manufacturing or preparation, probably for things like APIs, and this time I mean active pharmaceutical ingredients. So we do have pH sensors. We've already covered upon it in this discussion when we were talking about measuring the pH in rumens. Of course, we do have pH sensors. Because somebody's trying to do in-line measurements, they're actually trying to measure in the sample itself, not, you know. So I think because we do have flow cells, I have a lot of confidence that actually our pH sensors are pretty good at being in the sample and essentially, even if the solution is flowing, they'll run fine. And something just occurred to me that obviously, if you're going to put sensors into solutions that are later on going to be used as a pharmaceutical, you've got to be thinking about the sort of materials. I think these pH sensors, the one I'll recommend should be okay with that. So I'll discuss that in a bit more detail. Again, we do have the OmniSense platform. It will the electronics and the connectivity all stays the same, but the form factor might have to change for these kind of applications. This is an old image that I put up, I don't know, maybe 2 to 4 years ago, I think. And I was just imagining how these people would use this. So you'd have a sample, and essentially you could be pumping that sample over the flow cell, and you could either be returning this sample back. You could be returning the sample, or you could actually just sending it to waste. We don't need, if you want to do a sort of online or near-line testing of pH, you can actually, you don't necessarily have to have the pH sensor completely in the primary flow stream. You could actually just be sampling it using a pump and microfluidic tubing. So I've sort of, I have thought about this in the past. So you can have your sample, you can have your pump, you can have your flow cell, you can have your pH sensor in there. And you probably, in some ways, best to send that to waste, because then you don't have to make this argument that something from the pH sensor has ended up contaminating the sample itself. It might make for an easier sort of regulatory approval path. Now, the reason I brought up this diagram was actually because of this idea here. I got these calibration solutions here, calibration solution 1, calibration solution 2. pH sensors can drift, but in order to kind of keep them within spec, you can do a periodic calibration. But also, I could have actually, these kind of multi-way valves, I think the gentleman who's inquired is more than capable of knowing this, that you could actually have more reservoirs. You could also have cleaning solutions, because I know that he's interested in cleaning in place and actually doing sterilization in place. Now, sterilization in place, I think, is a lot trickier. Sterilization in place can sometimes mean applying a pressure to solution and raising the temperature to like 120 degrees Celsius. I think that's a sort of an accepted sterilization in place. We've never brought our pH sensors to 120 degrees Celsius, so that would actually have to be tested whether they could withstand it. The one I recommend stands a chance, but we've not done it. So cleaning in place is actually, I think, is quite straightforward actually, because you can have cleaning solutions. And by switching the valve, you could actually draw cleaning solutions over the sensor. So I have less of a concern about cleaning in place, sterilization in place. We've not done that testing at 120 degrees Celsius part. So that would be the bit that would have to be essentially characterized. But a lot of what I'm talking about, if you follow one of the links earlier on, you'll find that we do a lot of work with flow cells and syringe pumps, etc. It's, let's say this is new application, but the engineering has been done in the past. I think I'm going to recommend, we got a couple of sensors up on the website. I'm recommending this one as opposed to this one, because this one is actually ceramic. So the materials I feel are kind of more inert, and they more likely to take the 120 degrees Celsius for doing a sterilization in place effort. You can ignore our pricing because this is low volume pricing. If this works, you can get extremely competitive pH sensors in pricing, but you have to do the science first, make sure it's working, and then talk about volume pricing. They were because of the application, there's a couple of things we need to talk about, and that one of them is aseptic manufacturing. So Zimmer Peacock standardly do not make our pH sensors in an aseptic environment, but we do make some other sensors for use as implantable sensors in aseptic conditions. So just so your FYI, the sensors probably should be made in an aseptic environment. We do have what they call Class 7 clean rooms, and actually we test for the biological material in those clean rooms. So we do something called, the units of measurement are called CFU, colony forming units, I think it's per milliliter, and we actually test our environment to make sure that we're within specification. And the fact is, when we are manufacturing like in an aseptic way, as you would imagine, these gowns and hoods are not for fun. This is generally, the way we actually have to operate here. So I don't have a problem with shipping sterile products. And then we can actually package them also in an aseptic environment as well. And we are ISO 13485, so we can be, we can have a quality chain to make sure that these things really are as sterile as they need to be. So I'm going to, what am I saying here? Yeah, one of the things I was thinking about a little bit was that you want to clean in place and you want to sterilize in place. I have a problem with the sterilization because there are definite definitions around approved sterilization techniques, and I don't definitively know all of them. I know ethylene oxide, I know sort of steam sterilization, I know gamma sterilization, I know e-beam sterilization. I don't know if it's an accepted technique to actually argue that, because you're sort of exposing the sensors to things like pH 9, maybe pH 10, that actually that's also a form of cleaning and sterilization. But I'd have to talk to, or our RAQA team would have to be involved in that to help answer that question. I don't have a problem with the idea of cleaning in place, sterilization in place. Sterilization means something, and you have to make sure that your sterilization process is a sort of accepted sterilization technique. Actually, I just want to change tack slightly and say, if you're using the kind of pH sensors that we're using, the pH sensor itself could be actually a disposable, and that will actually get you then away from the whole thing about sterilization in place. Because essentially what you would do is you do aseptic manufacturing, terminal sterilization. The person would put it into the sensing rig, they would use it, and then they would dispose of it, and that would get you away from sterilization in place concerns. I actually think that's a good business model, because then you're going to get away from this whole cross-contamination issue, and it also generates a new kind of revenue stream for you. In the pharmaceutical industry, people are, they're extremely risk averse, and of course they should be, and actually they quite like to not reuse things. I mean, I spent, a lot of time working in that industry myself. I do understand the carbon footprint concerns. I do understand that. But I think low risk wise, use and dispose is possibly the strategy. And we do have our Julie database. I think it could play a really important part of this workflow, because Julie has this capability of bringing data and actually sending data onto other people's systems using an API, an application program interface. I just want to discuss a bit something called API to API. So the Inquirer was actually in line with something I've been thinking about for quite a while. That in the pharmaceutical industry, they make a lot of molecules called APIs. These stand for active pharmaceutical ingredients. And at Zimmer Peacock, we describe ourselves as sensor to API, sensor to application programming interface. So it's a bit of a pun, but API to API. So a lot of APIs used in the pharmaceutical industry are sort of organic molecules like this. I know many of you will watch these webinars or have watched Zimmer Peacock speak in the past. You know this already, but this is capsaicin. Capsaicin can be used with our OmniSense platform, for example, or our FoodSense platform. I just want to stop for a second and just say, active pharmaceutical ingredients are often have electrochemical signatures, and they can actually be quite distinct, these electrochemical signatures. I'm just letting this one run at the moment, but whoops-a-daisy. What's happening is we're doing the voltammetry of this molecule, and we get these peaks in a positive and in a negative direction. And what you'll notice is that when we do the second, let's say, sweep here, we get a new peak. So this molecule has a distinctive fingerprint. Now, in the context of an active pharmaceutical ingredient in a saline solution or in a buffer or in some sort of solution that's maybe for injection or maybe just processing, there's going to be a very distinct fingerprint for that molecule, which I think we can actually detect on our technology platforms. We can actually send that to Julie, and then in Julie we can both store it and we can qualify it and we can quantify it. So the Inquirer was asking about pH sensors, and I have touched upon pH sensors, but I also want to say there's a bigger picture here, which is I've sort of called it API to API here, which is Essentially, these active pharmaceutical ingredients in the pharmaceutical industry have distinct fingerprints. We can gather those fingerprints both probably in real time and inline, send them to the cloud, save the data in the cloud, basically as part of their quality system in the pharmaceutical industry, and also to qualify and quantify that data. So they have a sort of chain of custodianship, let's say, that they actually know, you know, was the material there and at what concentration was it there, and you can do that in real time. I think using some of the technology stacks. So I will look at the time and move forward a little bit. I'm not even sure how long we've been going today, but number 5 now, hypervalue electrodes. So at Zimmer and Peacock, I genuinely believe this. Belief, but it's because we're the biggest users of our own electrodes at ZP. So if you're looking for a low-cost screen printed electrode, come to our hypervalue electrodes, drop down the menu here, and you will find that we've got extremely competitive pricing on this. So I do encourage it. Now, when people have problems with our electrodes, importantly as well, we'll also support it. So these people are having some problems. So we are working with different analytes with SPs, with these screen printed electrodes. We are facing issues with repeatability after cleaning. So is it recommended to reuse sensors after cleaning? It's not recommended. Now, I would think slightly different if you were suggesting gold, but this is carbon. Carbon screen printed electrodes, that carbon is actually quite soft. So I'm not really sure that you can actually reuse carbon electrodes. So for example, in our chili sensor, we have a modified carbon sensor. We don't reuse the chili sensors. We have to throw them away after use. So it's not really my recommendation to actually use them to use them, clean them, and reuse them. Can you provide us the data on sensor reproducibility? Yes, I can, and I will put that in a slide in a minute. And they're cleaning with sulfuric acid. So there's a bit of a red flag for me there because sulfuric acid. Now, I think what's happening here is that I respect the gentleman, that they're used to using a technology called glassy carbon electrodes. And I think they're basically taking some of the cleaning techniques that are using glassy carbon electrodes and bringing them to screen printed carbon electrodes. Glassy carbon electrodes are all very good for R&D, but the problem with them is you can never make a product using these kind of technologies. They're too expensive. So sulfuric acid does seem a big no-no to me. It's one hell of a corrosive chemical. So first of all, reproducibility. Zimmer and Peacock makes the world's best screen printed electrodes. The biggest users of ZP screen printed electrodes is Zimmer and Peacock. Anything we ship is being tested. So we would test them. Now this is what we're doing is we're testing we're going to the batch of screen printed electrodes. We're sampling from the beginning, middle, and end, and we're testing those electrodes then, and we test them in parallel. We have a lot of parallel potentiostats. We send the data to Julie. Julie, you can log on, get yourself an account with Julie, and we actually then measure the relative standard deviation. So what's the standard deviation? And it's actually less than 5% in this batch. So sometimes the reason that we're so fast when we're doing sensor development is because when people are doing sensor development, they're often doing it on top of electrodes that are extremely variable. So they get these variable results and they think, oh, there's something wrong with my science. No, it's the screen printed electrode that's the problem. With us, they're really reproducible, and actually, our reproducibility is better than 95%, which is not bad is extremely good for screen printed electrodes. Now let's go into the data itself. Now it's a bit hard sometimes because you just receive data and a bit of an email, and you sort of have to sort of figure it out. So bear with me if I get it wrong and you're the inquirer. Now the thing that I understand by looking at your data is this, that you have some signals here. I don't really understand the one on the right hand side, but I understand this one here. You have some signals here which look to me suspiciously like silver. Now, silver contamination on working electrodes is something that I know about. I've known this for 20 years, and we as Zimmer Peacock keep a very careful eye on this. But you have silver on your working electrode, and I think it's coming about because when you're putting maybe the solution of sulfuric acid on this and you're cycling, I think you're helping to solubilize the silver off the reference and counter electrode, and then you're plating on the working electrode. And the more times you cycle, I think you're building up more and more silver. So I think your cleaning, potentially in sulfuric acid, is um, helping to dissolve silver off the working encounter, and you end up detecting it on the working electrode. So um, we obviously, and I've said it previously, we would detect and look for that silver, before we actually sort of shipped it out. You've got a lot of silver going on here. And I generally think it's coming about because I think you're using sulfuric acid at some point on this electrode, and you're ending up dissolving the counter and reference. And you wouldn't have had it with the glassy carbon electrode, but obviously there you don't have the reference electrode on board. So I hope that at least helps to throw some insight onto what's going on here. So let me just do, and yet again, I do have some old data, and this is 2018, about silver contamination on working electrodes. And I have definitely seen other vendors who have shipped this stuff and it's got silver on the working electrodes, so be careful of that. Right, so it's been a long one today. I'm not even sure how long this has been going on for. Like I say, I apologize that we started early because, like I say, we are at the AACC. I'm in the US, different time zone, a lot going on. So we've started it early, and my apologies for that. Glucose sensors. I've given you a bit of technical data because you're in Turkey. It's probably worth reaching out to that company. It's even free samples are really hard in Turkey. Osmolarity for dry eye. Take home story is because there's a predicative device, you might be able to get it to market. Get it for regulatory approval for less than seven million US dollars, but there's a lot dependent on your skills and your team, as well as the contractors that you use. pH sensors for 5 years. My biggest concern is actually that pH sensor coming out down through the alimentary canal and out through the anus, in much less than 5 years. Sensors in the biopharmaceutical industry. I do think the form factor that we make our pH sensors, and the fact that you could actually have a business model where I think the form factor is good, and the form factor and the way we manufacture means you could end up with a business model where actually you weren't doing sterilization in place. I can understand you might have to do sterilization in place if you actually put the pH sensor sort of into the bioreactor and the whole bioreactor is sterilized. But at least having the idea that you weren't necessarily reusing the sensors so much might be a good solution. And question number 5 was about our Hypervalue electrodes, and I really hope that some of the insights that we've given there will help you today. I realize that it's not the best internet in this hotel, so I apologize if the video stopped a bit short today, so my apologies for that. I hope that it restarts a little bit, but otherwise. So in summary, thank you for attending the webinar today. I'm sorry about any internet connections that have kicked in, and I'm also sorry that we did it a little bit early because I happened to be away on a trip this week. All right, so if you have any questions from Zimmer Peacock, don't hesitate to reach out to us. Okay, thanks very much.